Okay, let's get started. So last week we landed about here. So we were talking about different types of solutions to record EEG. So we had the passive electrodes, which is the old style where your skin needs to be prepared by scratching away with a razor blade, actually, the dead skin cells, so that the electrodes, when placed on the scalp, could be in good contact. This was, of course, not so comfortable, as you can guess. But meanwhile, better solutions have been found, basically by introducing an amplifier around the electrode itself, so that the signals already could be boosted up before being sent through the wire that connects to the rest of the equipment. And that was basically a big step ahead. So no longer this kind of razor blade stuff needed to be done, but only just mounting it in the electrode cap, some gel, and that was basically it. More recently, also technology came about where active dry polymers are being used. These are actually carbon-based electrodes, and of which the tip only is covered with AgCl, and that picks up the biopotentials. And then you have all sorts of interesting solutions that like strapping the electrodes around the head and so on. So these are more recent evolutions. Also, getting rid of the cap is also a concern. So quite some time ago, IMEC developed this kind of brace or headset or diadem, if you want to call it. That time it was with Bristol electrodes, so what's the problem now? Okay. Good. So yeah, but these Bristol electrodes are pretty painful when mounted on the skin because they actually indent even your skin. It's pretty painful, and that was originally used for animals, for cattle, actually, to do electrocardiography with animals, like cattle. And so this could be this is another form of this kind of diadem. This is another diadem. This is a thing we already showed with the straps. And then you have companies, Emotive. So we are at slide number 7. Slide number 7. So for the Mac users of the planet, we are at slide 7. So you have also these kind of solutions where you can where you have actually wet electrodes. These are sponges that need to be made wet, and behind this sponge there is an electrode, gold-plated electrode. And yeah, on the bottom right, you have another example of a polymer electrode. But these things, I mean, I've read stuff about it because it is not giving good quality. So I'm moving to the next slide, number 8. Here it shows how the montage is done. So there are different ways of recording EEGs. So as you know, it's electrical signal, so you need 2 poles, the plus and the minus, if you like. So we need 2 poles, and yeah, it means 2 electrodes at least in order to record voltages. And this way, several montages have been used. So the so-called single reference montage, in which case your reference electrode is an earlobe clipped on your earlobe or earlobes, the mastoid, which is the bone behind the ear. And these are solutions that, of course, are more amenable to more clinical, more scientific environments. What is easier to do is a bipolar design, where you use the electrodes that you have already and actually look at voltage differences between electrode pairs. It's the bipolar case. So Then another one is a similar design. It's actually looking first at the average of all electrode voltages and using that one as a reference. So using the average as a reference. So so these are solutions that are, as I said, more amenable to these last two are solutions that are used by more commercial-oriented solutions. And yeah, the Mac users should download that software that I've indicated. If you don't do that, it's not my problem. I mean, stop communicating that. If as long as you don't download it, yeah, you're doomed. Okay, so stop bothering the others about that. Okay, so next slide is slide 9. You see the electrode placement that is currently considered as the international standard. So the 1020 system or even the 5 system. It actually depends on. It actually depends on the distance covered between the nasion, which is this little dimple here and above your nose, and the inion, which is actually at the very far end of your head. So that distance, that perimeter can be divided, and if it's divided in 10%, that's the distance between the electrodes. That's the 10 system. It's just 20%, and it's the 20 system, and so on. So depending on the number of electrodes that are placed, they are of course closer to each other, and then in cases where there are less electrodes. So this is what it specifies. And then there is a naming convention. You have the different types of lobes where you're recording from. So the frontal lobe goes from the central sulcus to the front, and then you have the occipital pole. You have the central around the central sulcus, the parietal, and so on. So depending on that, there have been some naming conventions of the electrodes, as you can see here. The frontal with an F, the central along this line here, the parietal with the P, and the occipital with the O. And then you have also a convention for the numbering. So on the left you have the odd numbers, and then on the right the even numbers. So this is the traditional setup. But of course, for some commercial solutions, there is a compromise in the electrodes often, and they don't follow that convention. Slide 10 now, you see that if you have recordings, you have a lot of problems with blinking artifacts. So if you blink with your eyes, this is muscular activity, then you can get these artifacts that can corrupt your EEG data. And of course, for the more frontal electrodes, this is more prevalent than for electrodes covering the back of your head. So these are artifacts, are techniques to deal with that. And then here in slide 11, we come to the rhythms that have been, by experience, being used, being labeled. And of course, the first one, as I said before, was the alpha. The alpha is the most prominent signal in the EEG. It's between 8 and 12 hertz, and it's actually a bit everywhere. The scalp, it can be recorded, and that was discovered by Hans Berger. And then, as you can assume, beta was the second one to be discovered, right? And indeed, it's true, and so on. So the letters of the alphabet indicate also a bit like historically how it has been discovered. And then, yeah, you see the location of these powers and these bands and what they signify. So you have, like, if a delta is a very low, very slow variation in the EEG amplitude, which is due to sleep, for example, like in infants, but could also be an indication in adults of coma. And then, alpha is the most prominent one, and it goes down when you open your eyes. This is a bit typical test to see if your EEG is setup is working. Open eyes, close eyes, and the alpha should evolve with that. So if you open your eyes, alpha goes down. If you close your eyes, alpha goes up. So the sensory stimulation of your brain that is inversely proportional to the alpha power.